Assalamu alaikum everyone and welcome to our molecular biology ASCP preparation series. The MCQs we'll discuss today and in the upcoming session have been carefully collected from several years of past exams to give you the best preparation possible. I'm Dr. Loai and I'm really excited to share my knowledge and experiences with all of you through this series. Alright, so let's dive in together. In our previous lectures, we covered nucleic acid isolation and manipulation of RNA DNA. So today we'll move on to the third section of molecular techniques, separation and detection. So welcome to the season. As we know, these molecular techniques make up about 30 to 35% of total molecular biology ASCP exams, all right? So now let's focus on the questions related to the gel techniques, including agarose and acrylamide gels, as well as the capillary electrophoresis by reviewing past molecular biology ASCP exams questions, okay? So let's start it. So the first question we have today, what is the primary purpose of gel electrophoresis? Actually, gel electrophoresis is used to separate DNA, RNA, or proteins based on their size and charge. So the correct answer here is to separate DNA fragments by size, all right? All right, the second question here. Migration distance on the gel is? The correct answer is inverse of the base pair size. So the migration rate of DNA fragments is inversely proportional to their size. I mean, smaller fragments with the large inverse size migrate faster through the gel. All right, let's move on. All right, next question. Which of the following is not a factor affecting the migration of DNA in agarose gel electrophoresis? The correct answer is nucleotide sequence of the DNA. As we know, the migration of DNA in agarose gel electrophoresis is influenced by the size, shape, and charge of DNA, but not its nucleotide sequence. All right? All right, the question here, what is the effect of increasing the agarose concentration in the gel? All right, the correct answer here is increase the resolution of small DNA fragments. I mean, higher agarose concentration create small spores, which improve the resolutions of the small DNA fragments. All right, let's move on. Okay, the question here, what is the role of glycerol in the loading dye? All right, the correct answer is to increase the density of the samples. As we know, glycerol increases the density of DNA samples, ensuring it sneaks into the wells of the gel. All right? All right, next question here. What is the purpose of loading dye in the gel electrophoresis? The correct answer is to add weight to the sample and track its progress. All right. As we know, loading dye increases the density to the samples, ensuring it sneaks, as I said, into the wells and also allow visualization of the sample progress during electrophoresis. All right. Which of the following is used to visualize DNA bands in a gross gel? correct answer is ethidium bromide. As we know, ethidium bromide intercalates with the DNA and fluorescent under UV lights, allowing visualization of DNA bands. Which of the following is the disadvantage of using ethidium bromide? Okay, the correct answer is the toxic and mutagenic. As we know, ethidium bromide is the potent mutagen and is the toxic. So it's requiring a careful handling and disposal. All right, the question here, what is the typical concentration range of agarose used for separating DNA fragments? Actually, agarose gel with a concentration between 0.5% to 2% are commonly used to separate DNA fragments of various sizes. All right, let's move on. Okay, the question here, which buffer is commonly used in agarose gel electrophoresis? The correct answer is TAE or tris acetate editor. All right. Actually, this buffer is commonly used in agarose gel electrophoresis to maintain the pH and ionic strength of the gel. Okay, let's move on. All right, the question here, which of the following factors can affect the resolution of DNA fragments in agarose gel electrophoresis? Actually, the resolution of DNA fragments in agarose gel electrophoresis can be influenced by gel concentration, voltage, and buffer composition. Okay, so the correct answer here is all of the above. Let's move on. Okay, let's see the question here. Which of the following is the true about the migration of DNA in agarose gel electrophoresis? All right, the correct answer is the smaller DNA fragments migrate faster than the larger ones. All right, let's move on. Let's see the question here. Which of the following is the common method for recovering DNA from agarose gel? The correct answer is gel extraction kit. All right, let's move on. All right, now we'll move to the another type of gel, acrylamide gel. All right, let's see the first question we have here in acrylamide gel. Which type of gel is commonly used for separating small DNA fragments? The correct answer is polyacrylamide. As we know, polyacrylamide gel are used for separating small DNA fragments due to their high resolution compared to the agarose gels. All right, let's move on. All right, the question here is, what is the role of acrylamide in a gel electrophoresis? The correct answer is to form the gel matrix. Actually, acrylamide polymerizes to form the gel matrix that separates molecules based on size. All right. All right, the next question here. Which reagent is used to initiate the polymerization of acrylamide? The The correct answer here is timid. Timid actually is used along with ammonium persulfate to initiate the polymerization of acrylamide. All right. All right. Next question here. What is the function of SDS in SDS page? All right, the correct answer is to denature proteins and impart a negative charge. As we know, SDS denature proteins and give them a uniform negative charge. So this allowing separation based on size. All right, let's move on. Please, if you found this content helpful, don't forget to like, share, and subscribe. Your supports mean the world to me and help me create more valuable content for you. Thank you for being part of this journey. Let's continue. All right, let's see the question here. What is the role of ammonium persulfate and polyacrylamide gel electrophoresis? The correct answer is to initiate gel polymerization. As we know, ammonium persulfate is used as the catalyst to initiate the polymerization of acrylamide and bisacrylamide. All right, let's move on. All right, next question here. What is the purpose of using stacking gel in SDS page? The correct answer is to concentrate the proteins into a thin band. As we know, stacking gel concentrate the protein into a thin band, ensuring they enter the dissolving gel simultaneously for better separation. Okay, let's move on. All right, the question here. What is the purpose of using resolving gel in SDS page? The correct answer to separate proteins based on the size. As we know, the resolving gel has a higher acrylamide concentration, which allow it to separate proteins based on their size. All right? All right. All right. The question here: which of the following is the common stain used for visualizing protein in the polyacrylamide gels? The correct answer is Comasi brilliant blue. All right. Comasi brilliant blue is the commonly used to stain proteins in polyacrylamide gels, making them visible. Okay. Let's move on. All right. All right. Next question here. What is the function of bisacrylamide in polyacrylamide gels? The correct answer to cross-link acrylamide molecules. As we know, bis-acrylamides act as the cross-linkers, forming a network with acrylamide that create the gel matrix. All right, let's move on. Okay, next question. Which of the following is the safety concern when working with the acrylamide? As we know, acrylamide is a neurotoxin and should be handled with the care to avoid exposure. All right, let's move. All right, the question here. What is the effect of increasing the acrylamide concentration in the gel? The correct answer is increases the resolution of small proteins. Okay, actually higher acrylamide concentration creates small pores which improve the resolution of small proteins. Let's move on. All right, which of the following is not a component of resolving gel in SDSPH? The correct answer is ethidium bromide. Ethidium bromide is not used in SDSPH. It's used for staining nucleic acid in agarose gels. All right. All right. What is the purpose of using a reducing agents like DDT or mercaptoethanol in SDSPH? The correct answer to reduce disulfide bond in proteins. All right. Actually, reducing agents like DDT or beta-mercaptoethanol break disulfide bonds, helping to denature the protein for better separation. Okay. Let's move on. All right. Let's see the question here. Which of the following factors can affect the resolution of protein bands in acrylamide gel electrophoresis? Actually, the resolution of protein bands can be affected by voltage applied, acrylamide concentration, and buffer composition. So the correct answer here is all of the above. All right, let's move on. All right, the question here. Which of the following is the common method for recovering proteins from the acrylamide gel? The correct answer is electroelutions. Actually, electroelution is the common method for recovering and purifying protein from acrylamide gels. All right, now let's move to the last part we have today, capillary gel electrophoresis. All right, the first question we have in this group, what is the primary principle behind capillary zone electrophoresis? Right, the correct answer is separation based on size and charge. Actually, capillary zone electrophoresis works by separating analytes inside the capillary based on their size and charge. So this technique allows for efficient separation of ions, small molecules, and proteins. All right, let's move on. Okay, let's see the next question here. What is introduced into the solution during capillary electrophoresis? All right, the correct answer is electrical charge. Actually, in capillary electrophoresis, electrical charge is applied to the solution, creating electrical field. This causes charged molecule to migrate through the capillary at different speeds based on their size and charge. All right, so this is allowing for efficient separation of molecules. All right, next question here. Which technique is commonly used for analyzing protein charge variance and assessing isoelectrical points? All right, the correct answer is capillary isoelectrical focusing. Okay, actually, capillary isoelectrical focusing is a powerful technique used to analyze protein charge variance and determine their isoelectrical points. Okay, it separates proteins based on their isoelectrical points by applying electrical field in a pH gradient. This causing each protein to migrate until it reaches a point where its the net charge is zero. All right, let's move on. All right, there is a question here. Which technique utilize a micellar solution to enhance separation of analytes in capillary electrophoresis? The correct answer is Messelas electrokinetic capillary chromatography. All right. This Messelas electrokinetic capillary chromatography enhances separation efficiency by using micelles, okay, which help improve the interaction between solute and the micellar phase. This allows for better separation of both charged and neutral molecules in a capillary electrophoresis system. All right, the question here. Which technique combines aspects of liquid chromatography and capillary electrophoresis? The correct answer is capillary electrochromatography. All right. Actually, this capillary electrochromatography is a hybrid technique that combines the stationary phase used in chromatography with electrophoretic mobility of capillary electrophoresis. All right. So this allows for improved separation efficiency and resolutions. So making it useful for analyzing complex mixture. Let's move on. Okay, the next question. Which technique focus on the migration of ions based on their concentration gradients? All right, the correct answer is capillary isotachophoresis. Actually, this capillary isotachophoresis separate ions based on their electrophoretic mobility within a concentration gradient. So this technique ensures efficient separation of charged species by allowing each ion to migrate at a uniform velocity within a respective zone. All right, let's move on. All right, next question here. In a chromatography method aimed at detecting base substitution, small deletion, or insertion in DNA, as well as identifying DNA polymorphism, which technique is primarily employed? All right, the correct answer is high performance liquid chromatography. Actually, high performance liquid chromatography is commonly used for detecting base substitution, small deletion, and insertion in DNA, as well as identifying DNA polymorphism. It works by separating DNA fragments based on their sequence-dependent difference in retention time, so making it effective method for mutation and polymorphism analysis. All right, let's move on. All right, the question here. What is the primary purpose of denaturing HPLC? The correct answer is identifying mutation in DNA sequence. Actually, these techniques involve denaturing DNA or protein and separating them based on the difference in stability, which can be influenced by mutation. So, it's commonly used to identify genetic variation by analyzing the molting behavior of DNA sequences, which can change due to mutation. All right. All right, that's all for today. I hope you enjoyed this lecture. Please don't forget to like, comment if you have any questions, share and subscribe to our channel to receive all the latest lecture. I also encourage everyone to take the test to share your question with us so that everyone can benefit. That's all. Salam.